Good afternoon, everyone. I am Veronica Lam from City University, Hong Kong, State Laboratory in Marine Pollutant. So today I'm going to present to you some of our study of benthic dinoflagellates in Hong Kong. The topic that I'm going to present today is the diversities of benthic dinoflagellates in Hong Kong water and the impact of environmental changes on the growth and toxicity of the local dinoflagellate species. So benthic dinoflagellates are small algae. They are unicellular. They live on different substrates. For example, the rocks and then the corals and so on. So in some years ago, we have done a comprehensive study on finding the benthic dinoflagellates in Hong Kong. And we collect the samples in the eastern side of Hong Kong, as you can see in this spot. And in these locations, we have found some different types of habitats, like the rocky habitat, the coral habitat, and the macroalgae bed. So there is basically 4 questions that we would like to answer. The first one is, what kind of benthic dinoflagellate that we can find in Hong Kong? And the second one is, are they toxic? What kind of toxin that they can produce? And the third one, how the temperature change affect them? And the fourth one is the ocean acidification affecting these benthic dinoflagellates. So there are 6 popular benthic dinoflagellate genera, including Gambidiscus, Fukuyua, Ostreopsis forcentrum, and Vidinium and Colia. The reasons why they are so popular is because that some of the species of these genera can actually produce toxin that can causing some of the poisoning events, seafood poisoning, and even fish killing event. So in Hong Kong, we have found the species of the benthic dinoflagellate from 5 of the benthic genera. So among these, we have found 19 species that belong to the benthic species. So along the way, we have found probably 3 of the putative new species, including Fukuyua and forcentrum. And after we have done some toxicity tests, we know that there are one species, Fukuyua species HK type 1, that is a toxic species. So in our lab, we have a quick testing platform to test out of all of the different pollutants and toxin, including some harmless microalgae, invertebrate, and marine fishes. So you can see that there are pictures with the stars. These are the testing subject that we test for our toxic algae. So here are some of our results. We have done brine stream and sea urchin larvae, marine madeca larvae. This kind of testing, we want to see the mortality after exposing the toxic algae extract for over 48 hours. And then we also have done one homolysis analysis using the local marine fishes. So in the end, we have found 12 of the species that is able to produce toxin. So we have done some toxic analysis, and then we can find that one species called Colia moliensis, it can produce 2 toxic compounds, OA analog and AZA analog. And for the Colia trabeculis and Fukuyua wrestleri, we have found one compound that has been recently reported. It's called 44-methylgamberoin. And this one is a water-soluble toxin. And moving to the temperature change, we have designed a series of the experiments to test out of 7 temperature from 16 degree to 28 degrees Celsius degree. And then the algae that we choose is the Colia. The reasons why we choose Colia is because this species of coolea is widely distributed in Hong Kong. So almost every site that we found can find coolea. So we think this is very good subjects to look at. So we expose the different coolea species to this temperature. Then we further look at the growth and photosynthesis. Then we extract the toxin, and then do some toxicity test, and then also the toxin compounds analysis. And so here's some of the results. Shows two coolea species. Coolea canariensis and then Coolea tropicalis. So these two species can be found in Hong Kong, but they actually dominate. Their dominance area is actually quite different. For canariensis is they like to tend to distribute it in more cooler areas and temperate areas, and for the tropicalis they like to stay in the tropical area. So for the cell concentration, Coolea canariensis, they like to stay, they grow better in 24 degree based on the cell density. But tropicalis, they like to grow more in 28 degrees. And for the cell pigment concentration and then the photosynthesis efficiency, they tend to increase with the temperature. What I would like to highlight is tropicalis. They seem to increase more sensitive with the higher temperature. And here are some more results. And we have, after we expose these algae with different temperature, we also check on their toxicity with the toxicity test. And Coolea canariensis, it seems like there's no significant toxicity that we can see, because you can see that we have done some brine stream analysis. There are basically not really much effect on the animals. But tropicalis, no matter what kind of what temperature that we have exposed it to, is still very toxic species. And for the gene expression, for later on we have studied this. And what I would like to highlight is that we compare the 24 degree to the 16 and 28. The reasons why we compare to 24 degree is because the 24 is exactly in the middle. So we like to see comparing to the most coolest and then the most hottest temperature. And then we can see that in the hottest temperature, they tend to have more regulated genes changes. And this is another Coolea species is called Coolea malaysiensis. This species is distributed from tropical to subtropical, sometimes even found in temperate area. So we have this test. We have worked on the Artemia larvae. It's 48-hour mortality analysis. So this is the LC50 curves number. You can see that from the 16 degree to 28, the number is dropping. It means that the higher of the temperature, the higher of the toxicity. And for further to look at the compounds that they produce, most of the compound, OA, AZA1, and AZA1 analog 1 and 2, basically has an increasing trend from 16 degree to 26 degree. Then altogether they drop at 28. So I think this is quite one of the quite interesting part that we will look at more in the future. And for the fourth question that we would like to answer is the pH change with the temperature. So we have designed another study with 3 temperatures from 16 to 28 and 3 CO2 level from 400 to 2000. The reasons why we set this number is because we have tested that it represents different pH level. So 2000 is around, I can give you 2000 is around 7.8. The pH is much more lower than what it used to be found in the seawater. So cell size, we test on the cells. It's quite interestingly, we found that the higher of the pH level, the smaller of the cell that we can find. And this one is to look at the photosynthesis efficiency. And you can see that the green lines in every species that we have tested, the 28 degrees, it has the highest photosynthesis efficiency. Even though it is not all of the situation is like this, but you can see that the decrease of the pH level is actually have a slightly increase of the photosynthesis efficiency, except this one. So we combine all these factors and run a test, and we find out that CT and CC are completely separate from each other. The reasons why is that CT is, we can see that it's from a tropical area, and CC is a temperate area. It's really, it's completely different based on where they distribute it. And then in the middle, Coolea malayensis, it's they also, it's like in the middle because it, you can see that it distributed in the middle of these two regions. So we further look at the gene expression, comparing 400 ppm and 4,000 ppm CO2. And we can see that Coolea tropicalis, they have a very very low response to these pH change. And then slightly, not slightly, it's higher in Coolea malayensis, and the highest in the Coolea californiensis. And then, so we can actually see that in tropical area, they can these cells are much more stable when there are pH change. So for summary, Hong Kong is the biodiversity hotspot for toxic benthic dinoflagellate. We have found 19 species that belong to benthic dinoflagellate, and then 12 of them is toxic, and these algae can produce lipid-soluble and water-soluble toxin. And there's still some long way for us to isolate more algae to look at some of the unknown toxic algae. With the rising temperature, the growth rate of toxic benthic dinoflagellate will differentiate based on their adaptability. The production of the algae pigments and photosynthetic ability shows an upward trend. The difference in gene expression was more obvious in the rising temperature. And toxicity and then the toxin content tend to be increased with the rising temperature. So with the combination effect of the high temperature and high CO2 level means a low pH, toxin producing species tend to have a smaller cell size. And in the photosynthetic efficiency of the toxin production species was enhanced. And there are different gene expression level between the low toxin species and then the toxin producer species. And species mainly distributed in warmer area, the Coolea tropicalis tend to be more stable in the gene expression with the changes of the pH. So this is the end of my sharing. I would like to take this chance to thank you for all of these big guys and for their guiding and their effort and time to put into the study. So thank you so much for your attention.